Let's discuss the mechanisms behind the pain-relieving effects of opioids. Located in the brain and dorsal horn of the spinal cord are opioid receptors that mediate the therapeutic analgesic effects and side effects of opioid medications. These receptors are located in many areas of the brain, including the thalamus, and in the periaqueductal gray and locus coeruleus, that are origination for descending inhibitory pathways. When these pathways are activated, the signals descend to the spinal cord to inhibit ascending pain signals and prevent them from reaching the brain. Opioids act as agonists at three main receptor types to exert their actions. Mu receptors are responsible for the majority of opioid effects, including but not limited to analgesia, which is the pain relieving effect, euphoria, sedation, respiratory depression, and increased peristalsis in the gut, leading to constipation. Delta receptors in the brain may have a hallucinogenic effect. They are also located in the gut and cause decreased gastrointestinal secretions. Kappa receptors mediate dysphoria by reducing dopamine release. The best understood mechanisms of opioid action are those that occur in the spinal cord. Remember, the pain signals are transmitted from the periphery to the CNS by way of lightly myelinated A delta fibers or non-myelinated C fibers, which make up the primary neuron. In the substantia gelatinosa of the spinal cord, the primary neuron synapses with a secondary neuron that carries the pain signals to the brain. Let's take a closer look at what happens in the substantia gelatinosa. Taking a closer look, note that the pain signal is transmitted from the primary neuron to the secondary neuron by way of excitatory neurotransmitters like substance P and glutamate being released from the primary neuron to bind the receptors on the secondary neuron, causing excitatory postsynaptic potentials in the membrane of the secondary neuron, leading to an action potential that can be sent along the secondary neuron to the brain. Activation of G-protein-coupled new receptors by opioids in the membrane of axon terminal cause closure of calcium channels, leading to less release of excitatory neurotransmitters like substance P and glutamate from the axon terminal of the primary neuron. Opioids also bind to opioid receptors in the membrane of the cell body or dendrites of secondary neurons and cause of potassium channels that also leads to hyperpolarization of the second neuron and a decrease in action potential frequency in a secondary neuron, causing less pain signals to be sent to the brain. So now you can see how opioids acting as agonists on new receptors in the substantia gelatinosa of the spinal cord leads to a disruption in pain signals, ultimately decreasing the intensity of the pain experience. Here is a diagram summarizing the actions of opioids in the substantia gelatinosa. This is Abigail June Parsacala reporting for the Pharmacodynamics of Opioids. Now let's hear Aldrin Pedrosa for the Pharmacokinetics.